Hello everyone. So from today we're going to be starting off with the past paper questions of respiration. So this is the first question that I've got right now, and so yeah, let's start off. Describe the importance of ATP in cells, giving two examples of processes in which it is used. So it's asking for importance and two examples. So we're gonna first things first, we do know that ATP is used to provide energy, especially for processes like active transport, protein synthesis, muscle contraction, and etc. There's a lot of these type of reactions that you can mention. You should be able to get marks for it. So now let's move on. Cells generate ATP by adding a phosphate group to ADP. During the complete oxidation of glucose, cells have two ways of doing this: substrate level phosphorylation and oxidative phosphorylation. So figures 2.1 and 2.2 are diagrams that show the main details of these two processes. Okay. So part B says state precisely where these two processes occur in the cell. Substrate level phosphorylation. So this takes place during, you should know that this takes place during glycolysis, and glycolysis takes place in the cytoplasm of the cell. And now oxidative phosphorylation. So this takes place in the inner membrane, or you could even write cristae of the mitochondrion. However, this term is more preferred because it's more specific, and although the marking scheme accepts both, but sometimes you gotta be more specific when they ask you a question. So part C states, compare the relative amounts of ATP produced by the two processes when a molecule of glucose is completely oxidized. So it's asking to make a comparison. So we do know that oxidative phosphorylation will produce more ATP, around 30 to 36 molecules of ATP are produced during oxidative phosphorylation. And then we also know that only 4 molecules of ATP are produced during substrate level phosphorylation. Okay, moving down. Only substrate level phosphorylation is possible in the absence of oxygen. Okay, now explain why oxidative phosphorylation is not possible in the absence of oxygen. So we also should be familiar with the fact that oxygen acts as the final proton and electron acceptor at the end of electron transfer chain. You can just write it as ETC. The examiner completely accepts it. At the end of electron transfer chain. And we also know that NADH is not oxidized in the absence of oxygen. And also, what else? What could be the third point? The proton gradient won't be then set up due to absence of oxygen. Okay, so that's the first question done. Now moving on to the second question. You should be familiar with the positions of the specialized features of the mitochondria. You should know where exactly all the processes takes place, which should make it much easier. We know that oxidative phosphorylation takes place in the cristae, and this Krebs cycle takes place in the matrix. So if you look at these two lines, this represents the cristae, and I could just label as this part 1, and Krebs cycle, since it's taking place at the matrix, so this is the matrix, and I can label this as part 2. Okay, now it says describe 2 ways in which the structure of the mitochondrion is adapted for oxidative phosphorylation. Now, we need to keep in mind that this is only these adaptations are only for oxidative phosphorylation. So, the inner membrane is folded to increase the surface area. That's point number 1. And we also know that the mitochondrion contains the enzyme ATP synthase. Okay, now question part C, it says explain how the lack of oxygen will affect the respiratory processes in the mitochondria. References to processes in the cytoplasm are not required, so you do not need to mention about glycolysis. What you do need to mention is stages of respiration after glycolysis would stop, and this is because no oxygen would be present at the end of electron transfer chain to form water. Okay, so yeah, that's pretty much it. And fewer molecules of ATP would be formed because the maximum chemical potential energy won't be yielded. Okay, so yeah, that's that. Now moving on to question number 3. Figure 1.1 shows the molecular structure of ATP. Part A says, describe the main structural features of the molecule. Okay, so whenever you're given a diagram of ATP, you should be able to recognize all the constituents. So I can easily say that this is the phosphate group. Okay. And this is the pentose sugar, which is ribose in this case. And these are the nucleotides, adenine. So yeah. So that's all we need to describe. So it has 3 phosphate groups. Okay, that's number 1. It has ribose as as the sugar. And lastly, we know that it has the nucleotide adenine. Okay, so part B says, explain how ATP is able to transfer energy in cells. So it is a small and soluble molecule that releases energy upon hydrolysis. each time it hydrolyzes a phosphate group is broken, which releases energy of around 30.5 kilojoule per mole. Okay, so state how ATP is synthesized in mitochondria. Now, this, you basically need to outline the process of oxidative phosphorylation over here. So what you need to do is, um, how would you describe it? Okay, starting off, we know that the hydrogen carriers, NAD and FAD, formed in the previous stages of respiration are moved to the matrix of the mitochondrion. Here, they have their hydrogen atoms removed and split into its constituent protons and electrons. So electrons are sent to the electron transfer chain, while the protons are pumped to the intermembrane and then passively diffuse. Sorry, not passively diffuse, but rather I should use the term passively move back to the matrix, but this time they pass through the enzyme ATP synthase to form ATP from ADP in a process called chemiosmosis. So yeah, here are the few small questions that I want to go through, and I will be going through a lot more questions, so stay tuned.